Well, thank you for letting us fill in for this slot. So as she mentioned, I'm the co-founder of Ventrix, which is a local San Diego startup. So what Ventrix is working on is developing innovative therapeutic biomaterials for treating cardiovascular disease. So our initial product is Ventri Gel, which is an injectable natural extracellular matrix-derived scaffold, which I'll tell you more about. So for our team, as I mentioned, I'm the co-founder, so my day job is actually as a professor of bioengineering at UC San Diego. So I've been working in the field of injectable materials for cardiac repair, basically from the beginning, so starting about 15 years ago. And my lab takes an engineering design-based approach to develop new injectable materials for cardiac repair. And so this led to the development of the first cardiac-specific injectable material that we also designed to deliver via catheter, and that led to basically the formation of Ventrix and the Ventri Gel technology. So I co-founded the company with Adam Kenzie, who's the CEO, and as you can see, we have a relatively small and lean team with also additional expertise in clinical trials and business, specifically in the cardiac space. So for those of you who attended the cardiac session yesterday, you heard, of course, heart attacks and heart failure are a huge problem in this country, rest of the Western world. So every year alone in the U.S., there's about 700,000 people or 700,000 heart attacks or myocardial infarctions. 40% of those go on to develop heart failure, and at every given point in the U.S., there's about 2.5 million patients who are experiencing heart failure post-myocardial infarction. So, of course, this is a huge burden on the healthcare system, estimated to be about $26 billion. So there's a big need to develop new therapies to treat post-myocardial infarction patients and those patients that have already gone into heart failure. So when someone has a heart attack, there's a series of events that leads to that patient going into heart failure. So what I think most people have focused on when they're trying to design new therapeutics is that initial event that you see there, which is cell death. So you have a blockage of a coronary artery that leads to death of the cardiomyocytes and other cells in that infarct area. And that does help lead to progression to heart failure. But something else that occurs that has been largely ignored is you also lose the extracellular matrix, so the natural scaffolding that surrounds our cells that provides appropriate chemical as well as mechanical and structural cues for proper cell function. That's also lost because of degradation through enzymes that are upregulated after an infarct. So it's really this combination of cells and loss of the extracellular matrix that leads to this downward spiral to heart failure. So because the heart isn't very regenerative, that normal extracellular matrix is actually replaced by scar tissue. So a very abnormal environment that is mainly collagen-based tissue and really doesn't provide the appropriate cues for repair or regeneration. And that leads to left ventricular remodeling, which involves the left ventricle dilating and eventually leading to dysfunction and heart failure. So instead of taking the approach of trying to replace these dead cells, we decided to take a slightly different approach, which is focusing on that loss of the extracellular matrix. So if you're trying to deliver cells without having those normal cues, or to try to stimulate endogenous regeneration without those normal extracellular matrix cues, normally you would have scar tissue. I don't think you would expect that you would really get natural regeneration or repair, or proper regeneration or repair. So we focused on designing a material that would mimic that cardiac extracellular matrix that was lost to provide those natural cues for cardiac repair and regeneration. So what Ventrix's lead product is Ventri Gel. This is an injectable hydrogel that's cardiac specific. It's derived from porcine hearts that are decellularized to leave behind just the extracellular matrix, so that natural scaffolding retains all of the biochemical complexity of the natural extracellular matrix. And we process that in order to have it deliverable via catheter. So we can deliver it via transit transendocardial injections through many of the typical cardiac injection catheters. And then once it is injected into the tissue, which you see there in the middle image, that liquid reassembles into a porous and fibrous scaffold that's very reminiscent of the original structure of the cardiac extracellular matrix, which you can see there in the inset image, so which is when the myocardium has been decellularized, but before we've done the processing. So you have this liquid material, can go through a catheter, and then sets up into this new scaffolding for endogenous progenitors, blood vessels, et cetera, to migrate into to help repair the heart. So in terms of some distinct advantages, it is off-the-shelf, is an off-the-shelf product. So you can see the vial there. It comes as a lyophilized cake that can just be rehydrated with sterile water and then injected. And because it is a biomaterial as opposed to a living product, it's relatively inexpensive to manufacture, so it's actually about 2 orders of magnitude cheaper than typical cell therapy products. But as you'll see, we have still seen significant improvements in cardiac function with just the material. So we've done extensive preclinical testing, both in rat models as well as pig models of both subacute as well as chronic myocardial infarction. This slide here just shows you some of the data from our porcine subacute model that was published in Science Translational Medicine. As you can see on the left, looking at ejection fraction, so we infarcted the animals, and then 2 weeks later, you can see the ejection fraction declines as expected. And then taking the animals out to 3 months post-injection, controls continued to decline, whereas we got significant improvements in cardiac function by delivering Ventri Gel alone, again using that transendocardial injection catheter. We use the MyoStar catheter in this case. And then on the bottom graphs, you can see changes in diastolic volume as well as changes in systolic volume, showing decreases in both, particularly strong decreases in systolic volume. And we also saw increases in regional function, not just global function, which a lot of people look at just shaping the heart, but in fact improving contractility. And the reason we think this was occurring is if you look on the right, this is some of the histology images that are trichrome stain images. So we have red is cardiac muscle, blue is scar tissue, and specifically at the endocardium where we were doing those injections, we saw significant increases in a cardiac muscle, which is the graph on the left of that box. And then we also saw, so significant increases in cardiac muscle, and then if you look at the scar content, so the collagen content of the infarct, that was significantly less. So essentially greater cardiac muscle, less scar tissue that led to significant improvements in cardiac function. And we've also seen very similar results more recently in a chronic MI model as well. So in addition to extensive proof of function data, we also have extensive preclinical studies on mechanism of action. So as I showed you, the material initially sets up into this nice porous and fibrous architecture, this new scaffold that acts as a new physical home for cells to migrate into. And so we've done a lot of analysis. This is showing some example from our whole transcript array analysis, looking at what are the pathways that are affected when those cells migrate into the material. So we saw that actually you get whole genome-wide shifts when you inject Ventri Gel compared to saline injections. And the key pathways that were regulated were heart development, blood vessel development, altered inflammation, so shifting towards more pro-remodeling versus inflammatory environment, decreases in cell death, and also shifts in metabolism, so indicating we're really kind of kickstarting a pro-regenerative environment in the infarct area. And then in addition to the array data, we've also looked at a lot of histology as well as in vitro data. So we know that the material does increase neovascularization, but also it specifically affects cardiomyocytes. So we've seen indicators of a new cardiac muscle, so the increases in cardiac muscle that I showed you, but also upregulation of many early cardiac transcription factors, indicating potential cardiomyogenesis, so new formation of muscle. Also showed decreases in cardiomyocyte apoptosis, so after an infarct and all the way actually till a patient goes through heart failure and even spikes up again, you have apoptosis going on, and so we decrease that. And lastly, we also see shifts in cardiomyocyte metabolism, more towards an oxidative phosphorylation metabolism, which is more indicative of a normal, healthy heart versus infarct glycolytic metabolism. Ventrix now has an ongoing clinical trial in post-myocardial infarction patients. So it's a phase 1 study to look at the safety and feasibility of Ventri Gel in patients between 60 days to 3 years post-myocardial infarction. We are delivering this via the MyoStar catheter-based approach. And we're planning on 18 subjects. So far, we've treated we have follow-up data, complete follow-up data on 2 patients, and both did show signs of improved cardiac function. And we are, in addition to all the safety endpoints, we're also using cardiac MRI for assessing cardiac function. So for Ventrix's development plan, we plan by next year to complete that phase 1 study that I just showed you to demonstrate safety and also look for trends in efficacy. Then plan to complete phase 2 and phase 3 trials in the US. So interestingly, because it's a biomaterial, we have a unique opportunity in that because it acts initially as a physical scaffold for cells, it's actually regulated as a device in Europe. So in the US, it is a biologic, which will require the phase 1, 2, and 3 studies. But since it is a device in Europe, we do anticipate registering for a CE mark at the end of the phase 2 study, and then can be on the market in Europe earlier than the US. So with that, I'd like to thank you for your attention. I'd be happy to answer any questions at the break, or feel free to reach out to us. Thank you.